Yeah.
So there was a theory at one point in time that allergens that were in lotions that people used on baby's skin could have been causing some of the sensitization to those allergens. And so I'm just wondering, like, what is the view on that topic or theory?
Yeah, so the question was that there was a theory at one point, and actually there's data that I'll tell you about, that some of the lotions that mothers put on babies contained allergens, and children were getting sensitized to the skin, and that was leading to the whatever allergy they had, and that's absolutely the case. I didn't tell you this in the discussion about the leaf peanut allergy prevention study, but we think that most kids who are sensitized to peanut get sensitized through the skin, not by oral exposure, because kids often react to peanut on the first time they eat it, so they hadn't had prior oral exposure. And one of the ways we enriched for children at risk for peanut allergy development was that they had to have either egg allergy by the time they enrolled in the study or eczema, atopic dermatitis. And kids who have eczema have a broken skin barrier, and they have, in many cases, a genetic defect in one of the proteins that holds the skin cells together. So that opens up these channels where a peanut allergen could get in. And that's the kind of background theory about why eczema leads to other allergies, in particular peanut. But you asked about lotions, and it turns out that especially in the UK, but also here, maybe 15, 20 years ago, some of the lotions that parents were putting on babies, and it was the babies with eczema who, the parents want to put the lotions on to moisturize the skin. These had peanut oil as one of the components of the lotion. And there was enough peanut protein antigen in the peanut oil to get into the skin and sensitize these babies. And the odds ratio, if you had eczema and you got one of these peanut oil containing lotions versus kids with eczema who didn't get the peanut oil containing lotion, the odds ratio for developing peanut allergy was about 6-fold. And so that was a kind of nice, clean, retrospective experiment to nail that down. And that's why many other approaches to preventing peanut allergy or other food allergies now are going to explore preservation of the skin barrier. Other things people are doing, putting emoluments on that will serve the same purpose people are hoping to do with peanut oils, but not contain allergens.
Did that change how the cosmetics and industry selected ingredients to put in lotions or products used on infants and children?
Certainly for infants. As far as the overall cosmetic industry goes, I can't say. Yeah.
Thank you so much for talking today. I was really intrigued about your slides concerning adjuvants, and I was curious about what challenges that state faces in the wake of all these anti-vaxxers and the opinions that the general population might have towards vaccination versus folks who work here are a bit more in the know and know how safe and how necessary it is. So I'm just curious if that plays a role in some of the challenges that your division faces.
Yeah, it does, and both scientifically and from a regulatory perspective and from the public perceptions that you alluded to. And for the public perceptions, we don't have to deal with that so much as the Institute director does. Tony gets these questions when he goes to Congress or the lay press, But it's even more so, it's an FDA issue, I think. It's not been a huge issue for our institute. But scientifically, it's really intriguing because for many years, before we had all these new adjuvants in development, so probably about 25 years ago, a very famous immunologist named Charlie Janeway hypothesized that a whole set of receptors on immune cells might exist, and they would recognize things that we now understand are adjuvants, and they were necessary for generating an immune response. If he just gave the protein antigen to an animal, many times the animal wouldn't develop any immune response. It was kind of silent. And what he said was, it's the immunologist's dirty little secret that we have to use things like Freund's adjuvant, which you can't use in humans because it's too toxic, but we use it in animals all the time. And he said, these adjuvants they mimic something in the bacteria that triggers the immune response when we get an infection. And it turned out, a decade later or so, people found these receptors on the immune cells and identified exactly what bacterial products bind to them. Endotoxin is one, some of these bacterial DNAs are others, and with high specificity, this binding occurs and activating the immune response. One of the things that your question prompts me to tell you, though, is that for many years, the dogma was that these adjuvants are so reactogenic that to get a good vaccine response, you have to have what we call reactogenicity, which is inflammation, pain. The babies or adults say that vaccine hurt, or they get a fever. For many, many years, the whooping cough vaccine was really effective, but it caused febrile seizures in babies, other problems that 30 years ago, the Institute began a big push to develop the modern pertussis vaccine, which doesn't contain all the other material that the whole virus contains, whole bacteria contains. And with that, we produced a much safer vaccine, but one that's not as immunogenic, and probably accounts now for the waning immunity I referred to with pertussis, why years ago kids got the pertussis series and they were protected through teenage years, and even young adult mothers were protected, but now that immunity wanes. And so the dogma was that to get a good immune response, you needed this reactogenicity. One of the things we're finding with the adjuvant program is that with kind of more modern technologies and approaches and synthetic chemistry, we can modify some of these adjuvants and push the immune response down pathways that produce just as much antibody, but are not reactogenic at all. We have other adjuvant candidates that are very strong antibody inducers, but they're actually anti-inflammatory. So you could give one of those anti-inflammatory adjuvants in combination with a reactogenic adjuvant and generate a particularly desirable immune response, and maybe reduce the dose of the reactogenic component to a point where it's easily tolerated. So in terms of regulatory issues, yeah, we faced a lot of concern because before we could show that the reactogenicity was dissociable from immunogenicity and adjuvant activity, there is a concern that giving adjuvants, especially to young children, would cause things like autoimmune diseases. We would activate the immune system in a way that could become harmful and be very dangerous. So hopefully that's not going to be the case, but it means that these new adjuvants get licensed carefully and slowly. Adults first, then teenagers, babies last, of course. Yeah.
So you had mentioned high consequence viral infections. So up in Frederick, we have the high containment units where we have all that bad stuff up there. Can you think of ways that we can work together better, or things that you can't get done down here that we can help you with up there, or?
I would say that most of those kinds of studies that would be done would be DMID or Vaccine Research Center or intramural studies. In date, we're not going to be doing studies of infectious disease challenge models or that kind of thing in humans. We might be doing them in animal models, and that might be an area of working together.
And we have some immunology labs up there. I don't understand any of the stuff that they do, but it seems pretty sophisticated.
Yeah.
I have a question about your adjuvants and animal models. Are the mice good models, or what's sort of the gold standard, and what predicts what will be a good adjuvant for humans?
Yeah, the mice are not a great model, but they're the best model we have to work at low cost. And the ways we get around the limitations of the mice are, first of all, we can put a humanized immune system in a mouse now. And it's not, fully human, but you can manipulate components of it well enough to get an idea of, it worked really nicely in this mouse strain, but will it have any potential for human? That's one thing. We also can work in vitro with cells from mice versus humans and see with exquisite detail which pathways are activated. And so you can make comparisons between the mouse and the human now that are informative. The other thing is with like the CpG DNA vaccines or adjuvants or RNA adjuvants. These are small pieces of DNA or RNA. They might be 10, 12 base pairs that activate intracellular or extracellular receptors on innate immune cells that are the targets for these and that start the adjuvant program within the cells. Because they're all synthetic DNA or RNA, you can change the structure. You can change the base order. You can change the structure with high precision and fidelity, and then you can compare them. You could do kind of a structure activity relationship analysis. What does these structural changes do in mouse cells, and what do the same structural changes do in human cells? So people are able to predict now, if you have a good adjuvant in mouse, will it be weaker or 10,000-fold more potent in human cells? And that's the goal, to find ones that are more potent in human cells but are not toxic. Yeah.
Great. Every couple of months I hear about the Hellman Underground and work that people are doing with introducing parasites to treat autoimmune disorders. I was wondering if you could talk about if the institute is working on anything like that, or really just generally about the hygiene hypothesis.
Yeah, okay. So the hygiene hypothesis goes back a few decades, and people started asking, why are we getting more allergy? Why are we getting more autoimmunity? And there are a few diseases where even though autoimmunity is hard to diagnose and can take a long time to develop, in adulthood, there are a few like type 1 diabetes of children, where the peak onset is, there's two peaks, one at about 6 years of age and one at about 12. So it's, and that's not a disease that you can easily miss as a clinician. So it was very clear that a couple of decades ago that we're starting to see increases in allergy and some autoimmune diseases. And people are saying, why? What's different? And the genetics of the population can't change that quickly. Environmental exposures certainly could. And one of the big, environmental changes that we have as a society is we have clean water. We didn't have that 100 years ago. 50 years ago we did, but 100 years ago, clean water wasn't so much the norm. We have prevented a huge number of childhood infectious diseases that could be educating and molding the immune response in a certain direction. And we have a variety of other kind of public health advances that have made our environment and our society cleaner. And that's why it's called the hygiene hypothesis. And the hypothesis is that the absence of all these stimuli that have educated the immune systems of babies and children for tens of thousands of years, we don't have the right set of stimuli anymore, and the immune system kind of goes off track and starts attacking itself, becoming more pro-inflammatory, either with autoimmune diseases or allergies. And so then people started looking for, okay, what are the data other than, these changes occurred? Can we find populations where the changes that have occurred are quite different from each other, and do they differ in prevalence of these diseases? So the best example is studies done in Europe about 10 or 12 years ago, showing that typical German family farm has the barn and the house connected. And the children will be walking into the barn, and the barn animals won't be walking into the house, but all of the exposure to manure and the dirt and endotoxin that is in the barn is wafted into the house. So a woman named Erica Vamudius started this study looking at people living in those typical farm settings versus more Western types of farms or urban children. And there are huge differences in the prevalence of asthma. The kids who grew up in the barn house setup had much, much lower rates of asthma. And then people started figuring out not only was that the case, but what components of their environment were accountable for that, and even going as far as finding genetic polymorphisms that would protect people or make people at greater risk, but the gene-environment interaction involved exposure to the farm products. So we know that that's the case in asthma. Another study published about 2 years ago replicated that in an interesting way. There are Amish and Hutterite communities that live very close to each other. They're geographically, within a few miles of each other. But the Hutterites have adopted kind of Western farming practices. They use tractors. They keep their farm animals separate from the houses. The adult farmers are exposed to the barn life, but the children are kept out of the barns. And the Amish do just the opposite. They don't use mechanized tractor, that kind of stuff. Their children are like the German kids I was telling you about. And the rates of asthma in those two populations are just as different. The Amish don't get asthma, the Hutterites do. And they're genetically similar too. So it's a very clean experiment. So that's the kind of background to the hygiene hypothesis. And we are supporting some of that work. We are doing other things that look at the human microbiome and the environmental microbiome as either triggers or protectors of development of allergy. We have some interesting studies, just like the peanut allergy story, where we had it backwards in terms of avoidance of the allergen in childhood. We know now that we always knew, or not always, but since I've been working here since the late 90s, that children in the inner cities have a very high risk of asthma development if they're sensitized to cockroach and rodent allergens that they have in their household environment. And they have, if you're sensitized to cockroach as a child and you are exposed to cockroach as a child, you have about fourfold the risk of needing an emergency hospital visit for asthma in a given year's time. We often assume from that it would be good to clean the household environment, eliminate cockroaches, all that. And if a child is already sensitized, meaning they're allergic, that's a good thing to do. You want to decrease their exposure. But what we learned in the last 2 to 3 years is that if you look at the children from birth, as opposed to these other studies that looked at 4 to 6-year-olds who are already sensitized, if you look at them at birth and you measure the amount of allergen exposure they have in their indoor environment, we do that by sampling dust and then measuring the allergens in the dust. There's a very direct relationship. The more allergen exposure you have to cockroach, rodent, and a few other pests, dog also, and cat, the more exposure you have, the lower your risk of developing asthma. And that was a surprise. It turns out the pets are probably doing something like the farm animals, bringing certain microbes into the household environment. In those dust samples where we measured allergen, we also measured the microbial taxa that are in those. And there are certain taxa now that we know are protective and certain taxa we know that probably stimulate the development of allergy. So we're getting at the question through the microbiome, not so much through the issue you brought up earlier, which was, should we be eating parasites and things, worms? But we have supported a couple of trials in inflammatory bowel disease where Trichuris worms were given to the research subjects. The company that was manufacturing those worms had a lot of problems in regulatory issues, and those trials had to be stopped, but unfortunately. But probably the benefit there, if there is one, comes from the same kind of principles, but it's harder to demonstrate.
Or an interest in getting them to eat worms.
Yeah.
That's good. Any other questions?
All right.
Quick one, just in terms of the importance of data and its reuse, can you share something about informatic programs?
Yeah, we have much more so than some of the other studies that the Institute supports. The studies in DATE, and especially the clinical trials now, are kind of viewed with a whole lot of mechanistic studies that look at the underlying immunology. Very different from a typical vaccine study, which now they're starting to do this in some vaccine studies, but typical vaccine study a few years ago would take serum, take blood, measure antibody in serum at baseline, give the vaccine, measure antibody 4 weeks, 8 weeks, and a year later. And that might be the extent of the kind of overall mechanistic studies. Now, much more happens in the context of those studies. But in DATE, we have always pushed to have a deep immunological understanding of what's going on. And much more so than any other organ system, it's possible through flow cytometry and the fact that you can draw immune cells out of the blood to do very, very detailed studies. We have all the reagents to characterize cells with exquisite detail. So we know what's happening over time. We know what's happening in different populations of cells. And now we have a lot of gene expression data as well. So we have made a real push to make that data public. And
with, reasonable success, but we could do better. Investigators don't always want to make their data public, even though the primary paper's been published. But we, a couple of years ago, through the Immune Tolerance Network, we developed something called TrialShare, which is a public database. Anybody can set up an account, and you can access the patient-level underlying data from any published clinical trial. So you can, in fact, if it's a online publication, you can click on the figure in the online paper and it will take you right to the underlying data in TrialShare if you already have an account. And we designed this so that people could get access to this data without some of the usual hurdles that academics or industry scientists face, which might be, okay, if you want access to the underlying data, you're going to have to submit a proposal. What are you going to do with it? It's going to have to be reviewed by an independent board. You're going to have ethics review. You're going to have to get IRB approvals. We said these are de-identified data. For those of you who are not aware of the distinction, when you have fully de-identified data, somebody working with that is not doing human clinical research. That's the HHS definition. So you don't need an IRB approval. You don't need ethics approvals. You don't need a lot of these things. So we could make the data available to a high school student. And we're finding that there are a lot of people kind of outside the usual sphere of academic biomedical research who are interested in these data. So if you want to set up a TrialShare account, you can do that and find a lot of interesting tools for data visualization. You can sort data sets, you can filter, you can rerun statistical programs on filtered data. Say, instead of looking at the whole cohort, I want to look at the females over age 40 in the cohort. You can kind of redo the data analysis. We also have a website called Import that houses clinical data sets and animal model data sets from any NIAID-funded investigator and a few others too, who want to make their data public. Those are typically not clinical trial data sets as opposed to the Immune Tolerance Network TrialShare data set. But again, you can go into there and download data. And the same applies. You don't need approvals. Just open an account. So I have a question. Which was better, the Bamba or the Trader Joe's? What? You like the Trader Joe's, Chase? All right. Thank you for coming.
Thank you, Dr. Edwards. Really appreciate it.